Neural entrainment is the synchronization of the neural activity to the periodicity of the sensory stimuli. This synchronization generates the steady-state evoked potential, that is, oscillations in the electroencephalogram phase locked to the sensory stimuli.
The classic interpretation of the amplitude of the steady-state evoked responses assumes a constant neural response phase locked to the stimulus plus a random background noise not related to the stimulus. The stereotypical response can be obtained averaging over repeated presentation of the stimulus.
This approach ignores the dynamics of the response, as in the case of the evoked potential adaptation elicited by prolonged exposure to the stimulus. In animal models, the auditory steady-state response generated in subcortical brain regions adapts to the continuous presentation of amplitude-modulated tones. In humans, it has been recently demonstrated that the power of the fundamental frequency of steady-state visual evoked potential is stationary only in 30% of the subjects.
When the focus of the research is the dynamics of the entrainment, we can assume that the temporal evolution of the response will be the same in different independent experimental runs. Therefore, the average of the signal in every epoch across independent runs will provide an accurate representation of the long-term dynamics of the oscillatory response.
Based on that assumption, we have developed a method to characterize the time evolution of a steady-state response. The method consists of acquiring several recordings of the same experimental condition. Following, instead of averaging subsequent epochs within the recordings, epochs which correspond to the same time window in the different recordings are averaged.
In this study,